Hello, my name is Nurshahana Betusahami and today I am going to present about leaf extraction from Ipomea pescapre as an anti-cancer. Ipomea pescapre, commonly known as fish morning glory or railroad vine. Fish morning glory can be found in subtropical and tropical regions around the world, including the West Indies, the Americas, Africa, Asia, Australia, and the Pacific Islands and Texas. It is also found in the United States along the Atlantic coast of Georgia and Florida, as well as the Gulf Coast from Florida to Texas. Its function is to aid in the stabilization of sand dunes and the prevention of coastal erosion. The there are also many countries that use this plant as their herbs medicine, such as in Nigeria, the dried leaves of the plant are used to treat arthritis, and in Indonesia, young leaves are boiled in coconut oil to treat sores, and also in Malaysia, they use the juice from this plant to cure jellyfish sting. From the research that I have made on these plants, it contains many bioactivities such as anti-cancer, anti-inflammatory, antioxidant, and antispasmodic activity. For anti-cancer, it revealed that Ipomea pescapre maternal extract was more effective against melanoma than aqueous and petroleum ether extracts, as well as Waras, which is fresh juice extracts but less effective than the carbazine. And for anti-inflammatory activity, the extracts of the leaves of Ipomea pescapre is a traditional remedy for inflammations that has been proven in the experimental models. For antioxidants activity, Ipomea pescapre has the ability to scavenge free radicals and is a variable source of antioxidants phytochemicals. And lastly, for antispasmodic activity due to the tolerance and potential adverse effects associated with current drugs. The antispasmodic effects of several medicine plants, including Ipomea pescapre, has been reported. Ipomea pescapre is a valuable medicinal plant and the leaf extract contains astringent, diuretic, and laxative properties. Secondary metabolites such as alkaloids, terpenes, flavonoids, tannins, coumarins, carbohydrates, phenols, saponins, and phlobotanins, and steroids were discovered in the phytochemical screenings of Ipomea pescapre extracts, and secondary metabolites with various chemical sulfates exhibiting a wide range of biological activities have been isolated from Ipomea pescapre. The objectives are to obtain the extract of leaves from Ipomea pescapre, to study the bioactive compounds present on the extraction of leaves of Ipomea pescapre, and to determine the anti-cancer activity on the extraction leaves from Ipomea pescapre. This is where I will get my sample, which is Ipomea pescapre, which is from UMT Beach. Okay, firstly, for methodology is the firstly sample were collected. Ipomea pescapre leaves, both greens and fresh, were collected from the UMT beach. Next, for leaf extractions, firstly, the Ipomea pescapre leaves were washed and cut into small pieces. Before extractions, they were shade dried, ground to powder, and stored at room temperature. 200 grams of powdered material were extracted sequentially using solvents with varying polarities, including hexanes, chloroform, ethyl acetate, methanol, and distilled water. Then, mechanical stirring at room temperature, which is 25 degree, was used for extractions. After filtering the samples in organic solvents with a butcher, funnel, and Watchman number 1 filter paper, the residue was extracted more than twice. And lastly, the aqueous extracts were then freeze-dried and the organic extracts were concentrated in a rotary evaporator under vacuum at 20 degree and stored at -20 degree for further experiments. Okay, to detect anti-cancer activity, the MTT colorimetric method was used to test the anti-proliferations of Ipomea pescapre extracts on human cells. The cells were seated in 96 wells placed at 37 degree and allowed to attach to the substrate overnight in a humidified atmosphere of 5% carbon dioxide and 95% air. Different concentrations of extract were added in triplicate wells and incubated for 24, 48, and 72 hours. Etoposide and dimethyl sulfoxides at 0.1% final concentrations were used as positive and negative controls respectively. Cells were incubated with a final concentrations of 0.1 mg per ml MTT of solutions after the specified times of extract exposure. Following a 4-hour incubation period, the culture medium was replaced with 100 L of dimethyl sulfoxides to dissolve the parmesan crystal. A multi-well plate reader was used to measure absorbance at 570 nanometer, and the percentage of viable cells was calculated. And lastly, the concentration of the extracts that caused a 50% reduction in cancer cells proliferations was referred to as the IC50, which was calculated using concentrations, response curves, and nonlinear regression analysis. This table shows how I start and will plan my project in the future. Okay, for the expected outcome, plants have played an important role in the development of several clinical useful anti-cancer drugs, and it has been confirmed that more than 60% of currently used anti-proliferative compounds are derived directly or indirectly from natural sources. And cancer treatment with herbs has been simple and effective method, and there is evidence that herbal medicines can modify and improve the conventional cancer treatment method, and the results were expected to detect the anti-cancer activity in the extractions of this from Ipomia pescapre. These are the references that I used for this project. That's all from me. Thank you.